Hello everyone, welcome to the ITMS Academy. Let's have start our second lecture. The topic for today is the biotechnology in food processing and preservation. Let's see the introduction for this topic. The main aim of this chapter is to highlight the progress of application of biotechnology in food processing and preservation. To produce a safe and high quality food, the process and manufacturing protocol used in the food processing must be of food grade, that is, free from the health hazards. The application of biotechnology varies from the development of hybrid plant and genetically modified disease-resistant mutant strain containing seeds for high-yielding superior quality plants. Biotechnology has a pivotal role in food processing sector, especially in cereal processing, fruit and vegetable processing, beverages, oils and fats, dairy, poultry and confectionery processings. One-third of the processed food of the world are fermented foods, either natural or international fermentation, adding microbial strains. Food undergoes fermentation by international inoculation or by natural fermentation, and eventually these desirable changes appear due to the fermentation by microorganisms or by their enzymes, flavors, fragrance, food, additives, and other value-added products. Processing assures the food security by minimizing waste production and to improve its quality and security. Coming to our next slide is the food processing. Food processing involves various unit operation and techniques to convert raw, perishable and inedible products to consumable form with enhanced quality and shelf life. Processing assures food security by minimizing waste production and reducing the food chain and increasing food availability and marketability. In each step of food processing, contamination may take place. In order to reduce this microbial contamination, biotech techniques are applied. Techniques applied in the assurance of the food safety focuses on the detection and monitoring of hazards, whether it will be biological, chemical or physical. Therefore, the definition of a safe food is the food which is chemically, physically, microbiologically free from any harmful material or the level of the contamination present in the food will not cause any harm to the public health. Let's see what are the methods to improve the quality of microbial strain. Microbial cultures are improved for use in food processing application by improving the quality of microorganisms and the yield of metabolites using mutagenesis, conjugation, and hybridization. Let's understand first of all these three words. What is the mutagenesis? Mutagenesis is the process by which the genetic information of an organism is changed, resulting in a mutation. It may occur spontaneously in nature or as a result of exposure to mutagens. It can also be achieved experimentally using laboratory procedures. The second term is the conjugation. It is the process by which one bacterium transfers genetic material to the another through direct contact. During conjugation, one bacterium serves as the donor of the genetic material and the other serves as the recipient. The third one is the hybridization. Hybridization is the process of producing a hybrid. It refers to the process by which producing offsprings by mating two parents from different varieties or species. Recombinant genetic engineering is the best known technique to alter the purified microbial strain related to the food fermentation. Genetically modified strains are applied in the manufacture of enzymes, vitamins, amino acids, and other fatty acids. Let's see what is the genetically modified plants. First of all, let's understand how they are produced. In general, there are two methods. First is the biologistic method, that is the particle gun method, or by agrobacterium tumefacient mediated transformation method. In biologistic or gene gun method, the gene is directly shot at the plant cell under high pressure. And in second method, in agrobacterium mediated transformation, a piece of DNA which infects a plant is integrated into the plant's chromosome through a tumor-inducing plasmid along with the genetically engineered strain. Few examples are given to generate vitamins, minerals, and essential nutrient-rich transgenic plants, in which vitamin-rich plants comes at first. Let's understand what is it. Vitamin-rich plants. Transgenic plants can be manufactured using knowledge of biotechnology with increased content of vitamins in certain crops. Vitamin A. Vitamin A is essential for vision and during the embryonic development and for homeostasis in adult. Transgenic rice is one of the examples in which expression gene for beta-carotene was incorporated. Vitamin C, also known as ascorbic acid, is an important antioxidant and cofactor for various enzymes. Vitamin C rich plants are the only dietary sources for vitamin C for humans. Vitamin A. Researchers have taken initiatives to grow vitamin E rich plants. Various methods are being employed for developing such. Secondly, the essential phytochemicals. Firstly, we should understand what does the word phytochemical means. Phytochemicals are the chemical compounds produced by plants. They are generally have biological activity that play a role in plant growth or defense against competitor pathogens or predators. Phytochemicals are precious for human nutrition. Indoles, isothiocyanates, and sulforaphanes for vegetables such as broccoli, allylic sulfide from onions and garlic, and isoflavonoid from soybeans are known as the plant phytochemicals. Two genes, IFS1 and IFS2, encoding for isoflavone synthase in soybean, are revealed and expressed in Arabidopsis thaliana to activate the synthesis of isoflavonoid genistein. Heading towards the enzyme. Enzymes occurs in all living organisms and catalyze biochemical reactions that are necessary to support life. They are commonly used in food processing, preservation, and raw ingredient manufacturing. The use of recombinant DNA technology has made it possible to manufacture novel enzymes that are tailored to specific food processing conditions. Alpha amylases with increased heat stability have been engineered for use in the production of high fructose corn syrups. Fourth one is the DNA vaccines. Genetically engineered DNA is introduced directly into the body to defend an animal against any disease where cell can produce an antigen resulting in a protective immunological response. DNA vaccines may have a number of potential advantages over conventional vaccines, including the ability to induce a wider range of immune response types. Let's see, our next topic is the bioengineered animals. Genetic engineering or bioengineering in animals can be defined as the deliberate change in the animal genome using the techniques of modern biotechnology. It provides various tools for improving animal welfare and health techniques such as artificial insemination, embryo transfer, in vitro fertilization, cloning, etc. are used to improve the genetic makeup of the animal. It provides various benefits such as healthier offspring, healthier and safe for food production from animals, consistent quality, disease resistance, etc. Nowadays, Four techniques are being used to fabricate bioengineered animals, such as firstly, the transfer of nuclear material, secondly, the microinjection, thirdly, the viral vector infection, fourthly, the transfer of embryonic stem cell. Let's see the applications of bioengineered animals. Talking about the first point is the transgenic dairy cattle for modified milk. Bovine milk is well known as ideal food human as it contains balanced amount of vitamins and minerals such as calcium and also good source of essential amino acids. Secondly, the increased muscle growth in cattle. Myostatin is a well-known growth differentiation factor A (GDFA), a member of transforming growth factor beta family, releases myocytes which inhibits myogenesis so decrease the muscle growth and differentiation. Third point is the transgenic swine with reduced fat content. Bovine somatotropin, a growth hormone, is formed in the pituitary gland of young cattle and is well known for accelerating the milk production in lactating cows. Recombinant bovine growth hormone (RBGH) causes decreased fat content in transgenic pigs. Next is the transgenic poultry, that is egg as a bioreactor. Exogenous proteins are introduced to satisfy the consumer demand of various proteins such as biopharmaceuticals, and it is produced with the help of egg, which was used as a bioreactor in egg white. Next is the bioengineered fish. Genetically modified fish have considerable potential to further increase the yield of fish farm but have prompted serious concern both in Europe and the USA about the possible environmental impact on wild species. Improving fish growth rate. To improve fish growth rate, fish can be cloned and recognized from various species by introducing fish growth hormones. Increasing antifreeze property in fish. Antifreeze proteins and antifreeze glycoprotein can be introduced in the plasma of many fish species to incorporate antifreeze property that protects from freezing at sub-zero temperature in the cold water. These protein inhibits the formation of ice crystal of fish serum by lowering its freezing temperature. Let's talk about the bioengineered microorganism. With superior bioengineering practice, it is possible to characterize the important food stream with high precision, isolation, and improvement of genes involved in the process of fermentation and transfer desirable traits between strains or even between different organisms. It has mainly two applications, that is, inhibition of pathogenic bacteria. During manufacturing of fermented food, to enhance the security, hygiene, and proficiency in the food industry, the major thrust is to maintain the originality of the inoculating bacterial culture and also particular protective cultures. Second is the natural sweetener produced by microorganisms. Microorganisms can be extensively used in manufacturing and improvement of food products along with flavor enhancer in fermented food manufacturing practices. Nowadays, many of the methods use synthetic chemical additives for flavoring of food products. Next, let's talk about the food preservation. Food preservation is the process through which minimizing the biochemical, physical, and nutritional damage of the food. It is estimated that large amount of food products are lost each year due to spoilage. In each step of food processing, contamination may take place. To reduce this microbial contamination, biotech techniques are applied. So there is some practical techniques used for food preservation. Recent year, many food industries represent immense role in food technology. Genetically modified plants and animals are used to enhance taste, shelf life, nutrition and quality of the food. Genetically modified yeast and bacteria are used to produce enzymes in food industry. Let's understand the biotechnology tools used for the food preservation. Talking about the first point that deals with the recombinant antifreeze protein. Antifreeze proteins are potent cryogenic protection agents for cryopreservation of the food and pharmaceutical materials. A food assessment expression and fermentation system for the production and secretion of high level of RAFP was developed. A novel recombinant type first antifreeze protein analog was produced and secreted by Lactococcus lactis and a food-grade microorganism of major commercial importance. Next is the use of high-pressure processing. It is a non-thermal and highly capable preservation technique. Basically, focuses on inactivating those bacteria which are responsible for food diseases. In this technique, high pressure, approx 100 to 1000 megapascal, is applied to inhibit the bacterial growth without disturbing texture, taste, nutrition, and mineral quality of the food. It is safe for both type of liquid as well as the solid products. The next is the bacteriocells. These are the complex protein added during the food processing. These proteins are active against food spoiling microbes. Next one is the chelating agent. Due to their binding ability with lipopolysaccharide layer of microbes, chelating agent helps bacteriocell protein to prevent microbial growth. Fermentation starter culture. In fermentation technique, complex material is converted into simple substances with the help of enzyme. In this process, lactic acid is produced which decreases the pH of medium due to acid medium and low water activity prevent biological growth in medium to ensure prolonged preservation of the food. The next one is the radiation. In this process, food products are exposed under the gamma radiation. Radiations cause metabolic changes to increase the shelf life. Let's talk about the products preserved through biotechnology. Talking about the first point, meat and seafood. Most of the world population is nutritionally depends on the meat and seafoods. The frozen meat treated with the antifreeze protein showed less drift loss, less protein loss, and high score on juiciness by sensory evaluation. Next is the milk and dairy product. Milk is a good source of calcium and mineral considering as a complete health drink. Current research indicates that act a carrier for pathogenic microbes. Pasteurization is generally used to decrease the microbial growth, although it will not applicable a complete sterile milk product. Hence, use of bacteriosense seems good approach to improve the safety and quality of the milk or dairy product. Coming towards the next point is the fermentation product. The genus Lactobacillus is a heterogeneous group of lactic acid bacteria with important implication in food fermentation. Consequently, lactobacilli have been used for decades in food preservation as a starter for dairy products, fermented vegetables, fish, sauces, as well as less inoculant. Lactobacilli have also been proposed as probiotics and microbial cell factories for the production of nutraceuticals. Let's talk about the fruits and vegetables. Soft overcoat of fruit and vegetables are responsible for damage its nutrient value. Controlled and modified atmospheric storage, active packaging, minimal processing, hurdle technology, forging technology, and gamma radiation are used for fruit and vegetable preservation. Thank you for being with us today. For more series, please subscribe our channel ITLS Academy.